less people in the waiting room. Welcome, everyone. Thank you for making the time to be here with us today. It's great to have Priya with us, who's going to talk about the Trackoscope in a minute. Before, I would like to, for those of you who are here for the first time, I would like to introduce the Open Neuroscience project and seminar series. So Open Neuroscience is a user-led repository of open source tools and open science tools for neurosciences, where everybody can submit their projects, and then we do a quick check just to see if it's not spam, and then the information about the project goes on the website. So if you go to open-neuroscience.com, you're going to find all of those projects. And one of the things we do is the seminar series where we invite the developers of tools and methods and protocols to talk about their projects and give them a chance to advertise them and also talk to the community and potential users and potential collaborators. This is all done in collaboration with Worldwide Series, which is a series of web streams that started mostly during the lockdowns and the pandemic, but has basically spread beyond neuroscience, and it's now in several different fields. And today we have the pleasure of having Priya with us. Priya is a mechanical engineering undergraduate at Georgia Institute of Technology at the BUMLA Lab. She's interested in robotics and frugal technology. And over the past year, she's been working in this lab to develop the Trackoscope, which is presenting today. She joined the lab, and this is really impressive. I mean, the fact that she developed this thing at a very early stage in her career is pretty impressive by itself, but even more so because she started developing this as part of her as she was still in high school, right? And so really impressive. Priya, I am really pleased to have you here with us. And the floor, and sorry, before I hand it over to you, just so people know, they can please write questions on the chat here and on YouTube as well. We're going to be monitoring those two, and we're going to have questions by the end of Priya's presentation. But by all means, if you have questions, just write them so that we can relay them to Priya by the end of her presentation. Priya, the floor is yours. Um, Please share your screen and talk to us about the Trackoscope. And thank you very much for being here again.
All right. Um, is uh, is it showing well?
Yeah, we can see your screen perfectly.
Perfect. All right. So, all right. Yeah. Thanks, Andre, for the introduction. Today I'll be talking about Trackoscope, which is a low-cost modular autonomous tracking microscope that we've been developing over the past few years here in the lab. So for a bit of background, microscopy today in like the classroom is typically conducted using like handheld microscopes, like the one shown here, that I'm sure like most people have seen. These microscopes are usually cost between $100 to $200 and are standard in like most science classrooms. However, like the downside is that typically only like one student at a time can like you view the image through the eyepiece, which kind of limits the collaborative observation. And then like for around double the price, you can get like digital microscopes. And while these are a step up, they still have like some limitations. And then some microscopes even have actuated stages, which allow for like controlled movement of a couple of centimeters in the X and Y directions. But even then, field of view is still quite limited, meaning you can't capture broader behaviors of organisms or like large samples effectively. And then, of course, at the higher end, research microscopes, which cost upwards of $1,000, provide far more advanced functions and like unique specializations, which enable discoveries in like various scientific fields. However, like their price tag makes them impractical for most educational or hobbyist settings. Overall, though, commercial microscopes are often expensive and provide limited versatility, whether it's like the high cost or narrow focus on what can be observed. They like constrained with students, researchers, or hobbyists can effectively study. But luckily, in the wonderful world of open hardware and accessible tools like 3D printers, there have been multiple innovations in the field of microscopy, which make microscopy more accessible and versatile for both like educational and research purposes. And I'd like to like highlight a few of these real quick. So one such example is like the Foldscope, which provides a magnification over like 100x for less than $10. And this is paper-based microscope is like an incredible frugal tool that opens microscopy to those who may not have access to traditional microscopes. And then there's also 3D printed mounts that transform like a regular smartphone into a mobile microscope, which allow for higher resolution digital microscopy on a budget. And then finally, there's like full 3D printed devices like the open flexure microscope, which bring commercial grade digital microscopy to the hands of many. And like this device, which has been featured in this lecture series, offers features like remote operation, motorized moving stages, which make it a pretty robust and affordable alternative for researchers. However, despite all of these great advances, none of these solutions fully address the challenge of observing motile organisms effectively in a versatile and cost-effective way. And so this is kind of where Trackoscope fits in, offering a low-cost open source solution designed like specifically for tracking moving organisms over an extended period. So Trackoscope is a low-cost and autonomous two-axis tracking microscope designed for observing microorganisms across a larger field of view than a traditional microscope. By combining a custom digital microscope with a linear actuation system, Trackoscope can effectively observe organisms over around a 300 square centimeter tracking area, so about the size of an A5 sheet of paper. This feature is particularly useful for observing organisms that move freely over larger areas. So the microscope is controlled by an Arduino microcontroller, which interfaces with two stepper motors. These motors are capable of precise control and can have speeds ranging from 4 millimeters to seconds to track fast-moving ciliates and can go all the way down to 100 microns per second for slower moving protozoa. This level of speed control makes Trackoscope adaptable to a wide variety of organisms, each with different patterns, movement patterns, and speeds. And then on the optical side, this setup uses a Raspberry Pi high quality camera and paired with a swappable objective lens. So this modular design allows users to switch between objectives for different levels of magnification and resolution. So for instance, you can get 9 micron resolution with a 10x objective lens and 35 micron resolution with a 4x objective lens. So this flexibility enables Trackoscope to capture detailed images of microorganisms while maintaining a broader field of view for overall tracking. Additionally, this design is both simple and modular. Most parts are like flat laser cut components and minimal 3D printed parts, which makes it easy to assemble in less than 30 minutes using common fasteners. This ease of assembly and modular construction also allows users to customize the setup to meet their specific research needs, from classroom demonstrations to lab experiments. The cost of Trackoscope is also a big advantage, ranging from $300 to $400, making it a lot more affordable than a majority of commercial tracking microscopes or microscopes in general. Once assembled, Trackoscope is pretty easy to operate. All you need to do is connect it to a laptop with a USB connection with the accompanying open source software to start tracking organisms automatically. So the tracking software for Trackoscope is run by a simple Python script, providing intuitive user interface for both control and data gathering. The UI is designed to be user-friendly, allowing researchers, educators, or students to easily interact with the system and capture important data on like organism movement. It has a couple of key features like the live video feed of the sample, the organism's current location, like in the history of like where they've traveled, and the option to like save the visual and positional data for further analysis. This positional data collected includes like timestamps, the organism's coordinates relative to their starting point, calculated velocities, and things like that. This data then allows like researchers to perform further analysis, such as like identifying movement patterns or studying behavior dynamics over extended periods. The software also features like manual controls through a virtual joystick, enabling users to directly control the actuated stage. Could be useful for finding specific organisms to track, manual record data, or focusing on a particular region of interest if automatic tracking is not needed. To make the tracking as versatile as possible, visual tracking is employed using OpenCV, which is a widely popular computer vision library. In this case, I used OpenCV's like Channel and Spatial Reliability Tracker, or CSRT, with an initial region of interest selection. As shown in the flowchart, the user starts their track by selecting their region of interest, or in other words, like the organism that they wish to track. The tracker then identifies the organism and its bounding box and proceeds to track its movement. CSRT is highly adaptable because of its discriminative correlation filter approach. It means that the tracker constantly updates and refines its model of the organism's appearance as it moves, which allows it to effectively track organisms that change shape or appearance, like amoebas, stentor, or like worm-like organisms such as Spirostomum. One of the advantages of CSRTs, the adaptability and efficiency. Unlike higher end tracking microscopes that require like pre-trained models to specifically identify and track a single organism, CSRT is versatile enough to handle a whole range of organisms without pre-training. This also means that it doesn't require as much computational power to run compared to organism-specific models, which can be more resource intensive. And then once the tracker identifies the organism's location, the software calculates the required movements for the linear actuator to keep the organism within the field of view. The system doesn't always attempt to center the organism perfectly at all times, which could lead to jerky movements. Instead, it defines like a center zone as shown in the flow chart. So this smooths out the motion and ensures that the video remains stable and fluid, especially for slower moving organisms that may stop, or for fast moving organisms that may shift direction suddenly. So this like combination of adaptability and movement smoothing allows for the long-term continuous tracking of a wide variety of organisms that we'll cover in a bit. So to highlight Trackoscope's versatility, we track a wide variety of organisms ranging from slow-moving protozoa to fast-moving ciliates and even some multicellular organisms. These observations showcase the system's ability to capture and analyze diverse behaviors across different species and movement patterns. So in this first video, we're observing Actinosphaerium, which is a slow-moving sea urchin-like predatory organism that uses axopodia, which are thin hair-like extensions to capture prey. In this particular video, we tracked Actinosphaerium over 5 hours, and in this time we see it ate paramecium, which demonstrates like the predator-prey interactions that it sees. Gathering this type of data manually over 5 hours would have definitely been incredibly tedious, requiring like constant observation and repeated small movements. But Trackoscope automates this process, which allows us to track the organism in real time and collect detailed movement data pretty effortlessly. So this kind of opens up new possibilities for studying long duration behaviors like the Actinosphaerium feeding, which were previously too time consuming to analyze. Next, we tracked amoebas, which Trackoscope revealed some fascinating behaviors for. So here we have uh, two 1 hour tracks playing side by side. On the left, we see an amoeba in a standard setup, which is where it's confined between a glass slide and a cover slip. And on the right, we've created a less conventional setup with the amoeba placed in a petri dish containing a thin layer of water. Typically, the setup isn't used because the slow-moving amoeba in a large petri dish isn't practical with traditional microscopes, which have like a limited field of view, and manually following the amoeba would introduce sloshing into the sample. However, with Trackoscope's ability to track by moving the microscope, we observed something interesting. So as expected, the amoeba in the slide setup moves around by extending its pseudopods, which is a common form of crawling movement. In contrast, the amoeba in the dish setup displayed swimming behavior at the air-water interface. Um, this method of movement was quite different. So rather than relying heavily on pseudopods, the amoeba glided across the surface with like minimal extensions of its pseudopods, which doubled or tripled the speed compared to like its crawling counterpart. Additionally, in the dish setup, the swimming amoeba managed to capture 4 paramecium within the 1 hour tracking period, as it like skimmed over the surface of the water, which demonstrates a more kind of effective hunting strategy in this particular environment. Um, it's exciting to see like observations like these obtained from like a somewhat unconventional setup, which kind of really exemplifies what Trackoscope unlocks. Um, Trackoscope is also able to keep up with faster moving ciliates as well. So to demonstrate this, we track both Berseria and Stentor. So on the left, we have Berseria, which is a large ciliate known for its speed. This organism can reach speeds of up to like 1.7 millimeters per second, which is around 11 body lengths per second, which is quite fast for an organism of that size. In the track, you can see Berseria like rapidly moving and darting around as it swims. Over the course of this track, like Berseria was observed like traveling very large distances, like actively hunting smaller organisms like the paramecium in the dish. Without Trackoscope, like tracking such a fast and dynamic organism for more than a few seconds would be almost impossible given how sporadic their movement is. And then on the right here is Stentor, which is known for its like morphological changes while swimming and feeding. In this track, we observe two primary shapes, a more spherical shape while it swims, and the trumpet shape it adopts while feeding. Stentor also displays complex behavior, such as using cilia to draw food particles towards its oral cavity. In this track, you can see like Stentor's behavior right now as it attaches itself to a surface with a holdfast while feeding. So both of these tracks further highlight the diverse locomotive strategies used by ciliates. Berseria's like rapid swimming, and then Stentor's dynamic shape changes really show like the versatility of Trackoscope in capturing high speed, complex movements and behaviors in ciliates that are typically difficult to track with conventional setups. Lastly, we tracked a few multicellular organisms to show what can be learned with these tracks. For this, we tracked brine shrimp and tardigrades. On the left, we have a track of a tardigrade, also known as a water bear. While tardigrades are slow movers, they have a unique gait when crawling across surfaces. In this track, you can observe the tripod gait they use to walk, coordinating their like small legs in an alternating pattern to maintain balance and traction. Tardigrades are particularly like interesting because they can survive in extreme environments like space, and their movements like definitely like vary with their environment. This particular track is a 6.5 hour track. Um, if I let it play out a little bit, you can notice that there's some periods of rest where the tardigrade would conserve energy before continuing on to move. Um, so over here right now, you can see it's moving. There's also other tardigrades in the sample as well that the tracker is still keeping on. So yeah, like Trackoscope allows for this long duration track where we can observe these like different behaviors of movement and periods of like rest over hours, giving a deeper understanding of their like locomotion and um, interactions with the environment with like the debris in the images. So you can see right now it's take like stopping. Um, on the right here, though, we have a track of brine shrimp, which are small, like aquatic crustaceans. Uh, these organisms are known for their rapid swimming and like kind of erratic movements as they navigate through the environment. In this video, you can see how a brine shrimp larva swims in like a series of bursts and circular patterns propelled by their rhythmic limb movement. With Trackoscope, we can observe this movement and their patterns over an extended area and kind of followed them without losing track of their quite fast and unpredictable paths over here. I'll quickly let this video play out. Yeah, you can see like it's spinning in a clockwise direction, which may indicate that maybe one of its limbs isn't as strong. And so these are some of like the conclusions you can draw while being able to track such an organism for an extended period of time. So, and quickly going back to the tardigrades, once we have collected tracking data, we can extract even more detailed information about their movement through post-processing. In this case, we've used Deep Lab Cut, which is a pose estimation tool that uses deep learning to track specific points on an organism's body. Here we use it to track the tardigrade's limbs, identifying key points such as the front, rear, and central legs. So you can quickly see in this video here in real time as like the tardigrade moves its legs up and down. Each leg, the point on it, moves along with it after training it on the data. So with these points can extract like velocity and position for each individual leg to visualize and analyze the tripod gait that tardigrades use for walking. So on the graph on the left, you can see like the velocity of each leg as it's plotted over time, which highlights the rhythmic coordination between the legs, even as the tardigrade moves over a smooth surface as the petri dish, where its legs tend to slip. So similar to this example, data from Trackoscope tracks like the positional data and the video data can be post-processed to extract detailed behavioral features for a variety of organisms, whether it's studying movement efficiency like hunting patterns, or like we saw in some of the previous videos, predator-prey like interactions. This kind of analysis offers a way to dive deeper into like data collected by Trackoscope and then extract more information about organism behavior. So kind of wrapping up overall, Trackoscope is quite capable of observing a wide range of organisms and gathering meaningful data, but beyond its specific design, Trackoscope is a modular, scalable, and customizable platform that can be adapted to meet the needs of both like students and researchers. So on the right, we have an example of a customized version of Trackoscope built using wood and 3D printed parts. And it also features a motorized Z stage for enhanced control. This showcases like the flexibility of the platform and how it can be easily modified based on the available resources or specific needs. And on the left, you can see a track of spirostomum captured using dark field microscopy. This example illustrates how Trackoscope um, can be integrated with different optical systems such as dark field, which provide users with the ability to choose like the best technique for their experiments and capture visual data that they would like to. These are kind of just a few examples of what's possible with Trackoscope's customization options. Um, the core mechanism is scalable, allowing the use of larger or smaller linear actuation components, and the Thorlabs mounting bracket in the setup and magnetic mount on the y-axis allow for easy swapping of like optical components such as lenses or cameras for either a more expensive or cheaper setup, giving users the freedom to modify the optical system based on their specific requirements. Finally, the platform's large tracking area and its ability to observe organisms without restrictive confinement open up endless possibilities for experiments from tracking swimming organisms in a pond like environment to conducting precision studies on a particular organism behavior. So overall, I'm pretty excited to see what people are able to do with this platform. And yeah, I'm glad to having to open up for questions that people may have.
Thank you very much, Priya. This was amazing. Yeah, so the floor is open for questions. While we wait for people to be a little bit less shy, do you mind if I ask you one?
Yeah, definitely.
Um, so this seems very portable, right? And because it's based on a Raspberry Pi, in principle, you could just have a power bank and power the whole
Mhm.
thing, right? Have you guys taken this to the field somewhere and deployed,
Um,
next to the ponds directly, or did you all get it in the lab?
we have not like deployed it outside, but we have created like some small setups uh, that are variations of this that are more portable. But yes, it can definitely be powered based off of a battery power. But like this specific design, like we've mainly only tested in lab.
Mhm. Okay, cool. Also, so another question is, I remember reading from the preprint, and I didn't manage to read the paper yet, so I don't know how much
Hmm.
things have evolved since then. But I remember you guys saying that you could do very well X and Y, but Z was something that you still wanted to implement, right?
Yes.
How does that, so the first question is, why was that more difficult, and what does it look like now? Did you manage to work on that, or are
Hmm.
you working on that?
Yeah,
Uh,
so in this actual picture that you can see on the screen with the kind of wooden setup, we did have like that motorized Z stage.
mhm,
So while this it did work with um, focusing in the Z-axis when stationary, it's a little bit of a harder problem to solve while everything is moving.
mhm,
So what we noticed was that when trying to do it with a moving track and moving all uh, axes at the same time, in order to do the focusing, you need to figure out which direction, like in the z-axis, the organism has moved, which involves moving the z-axis both up and down, which will take the image out of focus and will end up messing up the track. So
right.
unfortunately, we weren't quite able to figure out focusing while tracking, but we were
Uh-huh.
able to implement that like stationary focusing
Mhm.
um, so before the track. But once the track starts, the z-axis height does stay stationary.
Okay. Yeah, I mean, it sounds like a complicated enough problem,
Yeah.
right? Because you need to be fast in doing all three axes at the
Hmm.
same time. So Natasha is asking a question. She's saying thanks a lot for the talk.
Hmm.
Just wanted to ask if you know how many labeled frames were required to track the limbs on tardigrades on deep lab cuts.
Yeah. Yeah. So when I did the training for it, in total, I labeled around 50 to 100 frames for the
Okay.
track, and definitely had to do like a couple of iterations of refining some
Mhm.
of them. But those 50 frames were enough to map out the limbs for that small, like 5 to 10 second clip.
Okay, cool. So Natasha says that doesn't sound too bad, actually. Thank you.
Yeah.
Marcus, do you have a question? Do you want to unmute yourself?
Hi, yeah. Thank you for your talk, Priya. This is very cool. Um, those little were they shrimp things? Looked like they were moving really fast, and I was wondering if there's this thing seems to be able to move quite fast. Was there anything that this setup couldn't keep up with? Or do you know the top speed of this? Um, and I was also wondering what type of motors you guys use for this.
Yeah. Yeah, so I'll quickly answer the motors question. We are using NEMA 17 stepper motors. Those can go quite fast, and as well with like PWM or pulse width modulation, like we can control like the speed of it. Um, but so with this setup, I capped the speed at 4 millimeters per second, which was quite fast. And then with and using microstepping and um, introducing a ratio with the pulleys here, as you can see in this image, we were able to like have such a wide range of speed for the various organisms. And then to answer your question about were some organisms too fast, definitely were. Like the Berseria track that I showed previously was one of like the fastest organisms we um, recorded. And so while it was able to keep up with it for maybe like 10 minutes or so, if it did some extreme like pivot or movement, it kind of lost track of it. So that was one of the things with like the tracker was while it was pretty adaptable, if the organism moved very, very fast, it did have some difficulty keeping up with it.
Thank you very much.
Great. Okay, so I'm not seeing any other questions, but I do have one more, if you don't mind.
Mhm,
And this is more of, yeah, how was so the preprint came out, the paper came out. You
mhm,
mentioned when we were talking before the live feed started that you've been adapting and putting different modules on it.
mhm,
And so my question is, were these motivated by people who contacted the lab and you and wanted, whoa, I want to use this in my lab? So my
mhm.
question is more in the sense, have you guys gotten a lot of feedback from different users from different places, or what does it look like?
We've gotten like small bit of feedback from a couple of people who've used it, but like majority of it has been like driven by what we want to
Mhm.
kind of see. Like the dark field edition was from feedback during the peer review process of the paper, and other modifications were from uh, internal like needs from like the lab that we wanted to try to explore.
Mhm. Okay, that's pretty cool. Yeah, so I don't know if people have other questions. This is a time to ask them. Because we are, yeah, coming, I guess, if there are no more questions and if Priya doesn't want to add anything else
Mhm.
anymore, I think we can then thank Priya once more for her presentation. This was very nice. I really enjoyed it, hearing from you more about it. We as a lab, so the lab that we have here in Sussex, we're definitely going to look into, as I mentioned before, to try to replicate it for some of our things, and then definitely provide feedback and keep in touch. So yeah, so thank you very much. Thank you very much for everybody who joined us today. If nobody else has anything else to add, I would like to say thank you. And yeah, see you next time. Bye bye, everyone.